And translation, in its simplest definition, is moving from a nucleotide sequence into an amino acid sequence. Okay, so translation is changing from a nucleotide sequence on mRNA to an amino acid sequence that will form the primary structure of a protein. Okay, so let's draw out a very small mRNA strand here. So this is going to be mRNA. Okay, and on this strand of mRNA, there are going to be some nucleotides. Okay, so let's say the nucleotides on this strand are something like UUU, AUG, UAU, UUU, GAU, UUU, or something like that. Okay. So these get divided into segments of 3, right? And the segments of 3 are each called codons. So this is one codon, right? This is another codon next to it. This is another codon next to that. This is another codon next to that. You get the idea what I'm going for here. Another one next to that. And one next to that. Okay, so we can just count really simply here, and we can say 1, 2, 3, 4, 5, 6 codons. That's going to correspond to 6 different amino acids, right? But the big question is, how do we go from having just nucleotides here to having the correct amino acids go into those spots? And we rely on something that's called tRNA. Okay, so tRNA is made of RNA. Obviously, it's called tRNA, right? And it has a couple different parts to it, right? But it's kind of shaped like a T, like this. Okay, and on the inside, it's the way that it gets shaped like a T is that the complementary base pairs are hydrogen bonded on the inside of this particular RNA. So these are all bonded together here, and then on the end, there are nucleotides that are facing outward. Okay, so these nucleotides on the end that are facing outward, they are going to have what's called an anticodon on them, and that anticodon is going to correspond to the complementary base pair sequence of each of these little codons. So this one, let's say, is AUG, so the anticodon would be UAC. Okay, now each tRNA has that on one side, that's called the anticodon, and on the other side it's going to correspond to one particular amino acid. So we need to figure out which amino acid it corresponds to, and so we know that this is the anticodon, so it corresponds to this codon over here, AUG. So we need to check the chart to see which amino acid that corresponds to, right? So we'll check our chart here. We said AUG, right? AUG is methionine, right? You see that? Methionine right there, AUG. And so we could say that on this end, there's going to be an amino acid called methionine. Okay? Now I'm gonna leave it in this spot, but I just want to show you that this could then go over here and it could meet up with that codon, right? So that anticodon hydrogen bonds to this codon, and it would put methionine in this position right here, right? It doesn't put it here because that doesn't hydrogen bond. It doesn't put it here. Only puts it right here. Make sense? All right. So, tRNA is interesting then. So how does tRNA work? It has one particular anticodon here, and then it corresponds to one amino acid. That must mean there's a lot of different types of tRNA. And you're tempted to say, well, yeah, there's 20 amino acids, and therefore there are 20 different tRNAs. Does that seem logical? Right? If each tRNA holds on to one of these, then there has to be 20 of them. But in fact, that's far less than there are, because what we have to figure out is how many unique combinations of codons we can have up here. So if there are 4 nucleotides, right, and each one of them gets arranged into different 3 nucleotide sequences, we have to determine then how many total possibilities there are. I think the math behind that is you take 4 to the 3rd power. Yes, right? And that's going to tell you how many possible combinations there are, which comes out to something like 64. So if we look, there's actually 64 different types of tRNA on here, right? And so if we look at this, you can see this should correspond to 64 different types on here, right? This is 8, 16, 32. No, it's actually 128, right? So is it, how did I mess up my math? 8, 16, no, 8, 16, 32, 64. No, I was right. All right, just can't count. Counting is hard. So each of these things on here is a unique tRNA, right? So we're adding to our list of things that we can always assume are in plenty of concentration inside the cell. We can always assume that nucleotides have plenty of concentration inside the cell. We can always assume that hydrogen ions have plenty inside the cell. We can always assume that amino acids, there's plenty of them inside the cell. And we can always assume that tRNA, there's plenty of them inside the cell. Right? So the question, where do those tRNA come from? They come from the cytoplasm. They're just in there, right? Okay, so here's one type of tRNA. Next to this, let's draw another type of tRNA. Right, so we have the same type of general structure. Okay, so it's got the anticodon up here. And it's got the amino acid hooked on at the bottom. Okay, let's say this is the anticodon for UUU, so that would be AAA. So if we had this, it would go over here to AAA. And we go over here to UUU. And UUU is phenylalanine, PHE. And so we just draw on phenylalanine corresponds to that tRNA. Yeah?
Are we expected to memorize that?
Absolutely not. The table? No. Okay. So here are just 2 examples of tRNA that could go on this. Each tRNA has a couple different points on it that I want to talk about. The first point is this one at the top. Okay. This area is called the anticodon. Right, and so all these things make up tRNA. And tRNA is called transfer RNA, but its main job is to translate between the nucleotide sequence and the amino acid sequence. So these translate between a nucleotide sequence and an amino acid sequence. Okay, so how does it do that? It's got this anticodon thing. On one side there's an anticodon. And the anticodon is the complementary base pairs that correspond to the codon on the mRNA. Okay? And the second part is often referred to as an enzymatic hook, right? So down here, this could be an enzymatic hook. And the enzymatic hook is going to bind to just one type of amino acid, right? So we can say that every tRNA, every unique tRNA has an enzymatic hook that binds to just one amino acid. In other words, this tRNA up here at the top, the one that has phenylalanine on it, if it gets used and the phenylalanine comes off, it's going to just float around in the cytoplasm until another phenylalanine gets attached to it. It's not going to attach to a methionine or a glycine or anything like that. It's always going to have phenylalanine, right? And this one's always going to have methionine. Make sense so far? Okay, so let's do a quick recap of all the things that we know. Here's DNA, right? This DNA has been, it's been unwound here, so you can see this top end is not even showing, but the top end would be up here. And so it's unwound so that this is exposed, right? And there is a nucleotide sequence on here where each one has, a certain thing that can fit into, right? And then with this, the complementary base pairing can go on to create something like mRNA. So here is the mRNA that corresponds to this particular sequence of DNA, right? And you can see that all the complementary base pairs are in the correct spot. They're all fitting in nice and snug, right? And then once those are all complementary base paired, then RNA polymerase will come along and it will join together all these sugar phosphate backbones, so that now you just have one piece of mRNA. That's this thing. Now this is only showing 9 nucleotides. They're going to be significantly longer than that, like 1,000 nucleotides, but that would take a really large space, and so we have 9 here. Okay. This is the DNA. Obviously the DNA is going to be much larger, but you can see that the DNA is way too big. You can't have the DNA going out in the cytoplasm every time, so we make a small copy of it. We leave that stuff in the nucleus. This stuff goes out in the cytoplasm. Okay? So once this is in the cytoplasm, it is going to join up with tRNAs. So here's a tRNA, right? And this tRNA is going to fit in to this particular anticodon right here, right? Like so. And because it's fit into that particular anticodon, it is going to deliver its specific amino acid. So here's its specific amino acid. It delivers its specific amino acid to that one spot right there. And then the next tRNA will attach to this one, and it will deliver its specific amino acid to this spot, to the next spot over. And then this tRNA that attaches to here will deliver its specific amino acid to that spot, right? And what you're going to end up with then is a chain of 3 amino acids, starting with this gold one, and then whatever one is here, that'll be the next thing in the chain, and then that will be the next thing in the chain, and that forms the primary structure of the polypeptide, which can then be folded and stuff to turn into a protein. Make sense? Okay, so the process of allowing this to happen is kind of complex. We're going to talk about it over the course of 2 days. We're just going to cover the first step today. So the first step today that we're doing today is called initiation. So we can say that we have the first step, which is initiation. And what happens in initiation, there's a couple things, right? We'll start down here. So the first thing that happens in initiation is that a small ribosomal subunit attaches to the G cap of the mRNA. If that G cap is not exposed, then it will not have a small ribosomal subunit attached to it, and it won't get made. Okay, so we'll talk about this later, but there's an interesting little interaction here where we're finding more and more about this stuff called miRNA or microRNA, and microRNA's job, its main job, is to interfere with mRNA. Okay, and so what can happen is it can have a little small piece of RNA, maybe only 10 nucleotides long, and that will complementary base pair onto the G cap of certain mRNA. And because it's complementary base paired onto the G cap, it doesn't allow the ribosome to attach to it. And if the ribosome can't attach to it, then it doesn't get made into a protein. And so it can inhibit the creation of a protein just by having a couple of nucleotides bonded together. And so we're finding out that things like viruses have specific microRNA or interference RNA on them to prevent the body from fighting them off. So the body would produce a protein in order to fight them off, and so they produce a little strand of microRNA that interferes with the mRNA that the body would produce to fight off the virus, and then that virus or that protein can't get made, and then the virus gets to replicate and go about its way, right? So some pretty complex stuff here is going to be coming up, but now we kind of just got to get the basics down. So attaches to the G cap. Once it's attached to the G cap, that signals the first tRNA to attach. So tRNA that corresponds to the start codon attaches. So that tRNA that corresponds to the start codon is always the same. It's AUG, right? That's always the start codon. It always codes for methionine, right? And once that AUG codon is on there, that's going to signal the large ribosomal subunit to bind to the small one, right? Which kind of traps that first piece of tRNA in its spot and starts the process of protein synthesis. So we can say that the large ribosomal subunit binds to the small one, and begins process of translation. So we're going to do a little drawing, bigger drawing than we did up top. So here is strand of RNA, of mRNA. All right, so this one's got a lot of nucleotides on it. Let's say it's something like GGG, and then UAC, and then AUG, and then UUU, and AU, UGA, AAA, 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 AAA. Okay, so this is a fairly realistic interpretation of mRNA, right? Except for the fact that it's super short in the center in the actual gene portion. But on the end here, we've got our GCAP, and on this end, we've got our poly A tail, right? And so our small ribosomal subunit is going to attach to the GCAP, like that. Okay. Then our tRNA that corresponds to our start codon will attach. And so that would be UAC. We said that codes for methionine. And then we have our large ribosomal subunit that binds to the small one. And the large ribosomal subunit is going to be divided into 3 different parts. It's going to have what's called the A site, the P site, and the E site. And we'll write down what all these do tomorrow. But essentially what happens is new amino acids can enter the ribosome in the A site. So A for amino acid. The polypeptide, which is the chain of amino acids, is going to be stored in the P site. And then the E site is the exit site where tRNA that's had its amino acid removed from it can exit out of the ribosome. And this ribosome is going to travel down the mRNA this way, and that's called translocation. Nope, not translation, translocation. So it's translocating down this mRNA here, and it's going to do that until it gets to a stop codon. So here is our start codon, AUG, right? And then it's going to go all the way down until it gets to a stop codon, and I used the stop codon UGA, at least I thought that was a stop codon. UGA, yep, it is. So stop codon is here. So we go. This is the stop codon. So this is only going to make a 3 amino acid long protein, but you get the idea that if you just added more stuff in the center there, that would make a protein. Okay. Some cool stuff about this. There are some really long proteins that have to get produced in fairly large amounts. And so it's not just like one ribosome can attach to this thing. One ribosome could attach and then get like 15 codons down, and then have another ribosome attach and go, and 15 codons down, and another ribosome attaches. So there could be like 50 ribosomes on a single mRNA moving down. They call that a multi-ribosome or multi-ribosomal mRNA, where it's like being translated a whole bunch of times at once. And that's going to make a whole bunch of the proteins. And yeah. We said there are 3 sites. There's the A site, the P site, and the E site. The A site is where new amino acids are going to enter. P site is where the polypeptide chain is going to be held, and E site is the exit site. So how does a molecule of tRNA move from one site to another site? Well, in actuality, it doesn't. Okay? What happens is the ribosome moves. Right? And as the ribosome moves, these tRNA molecules are still bound to their anticodon, or the codon is bound to the anticodon, right? So AUG would be bound to UAC here. The ribosome moves down one spot, and that means whatever was in the A site will now be in the P site. Whatever was in the P site will now be in the E site, so on and so forth. Make sense? Okay, so let's talk about some vocab type stuff real quick. So if I have this tRNA and this tRNA, is it recording? Yeah. All right. Here's two tRNAs. Let's say they both code for the same thing. This one's going to be UUU, so AAA. Like so. And they both have their little enzymatic hook on the bottom like this. And I can look on the chart and see that UUU is up here. It codes for phenylalanine, right? So I can just put that on here like so. And let's put it on one of them. Okay, so this one has phenylalanine on it, this one does not. Okay, so what happens is, because this this particular tRNA is bound to a phenylalanine, we say that it's activated. It's an active form of tRNA, and specifically, since it's activated by an amino acid, we call it an active amino acid. Okay, this one that doesn't have an amino acid on it is called an inactive tRNA. Okay, inactive tRNA doesn't have an amino acid on it, and therefore it's not going to be bonding to these anticodons, or it's not going to be bonding its anticodon to the codon, and it's not going to be going into the ribosome. What do you think the inactive tRNA does? What's its goal at this point? Yeah, to become active. So it's got this little enzymatic hook here. It's just going to be floating around in the cytoplasm looking for a phenylalanine, and when it finds a phenylalanine, it's going to grab onto it. That's going to make it an active amino acid, and that active amino acid can then be used in the process of translation. Make sense? Okay, so let's talk about this second step here. The second step is called elongation. And the first part of elongation is when an active amino acid enters through the A site of the large ribosomal subunit. So an active amino acid will enter the A site of the large ribosomal subunit. Thank you. And how do we determine which active amino acid is going to go into that A site? Can just any active amino acid go in there to this particular A site? How do we figure out which one's going to go in? What? Whatever the what is? Yeah. Yeah, clubs can go.
Whatever is attached to the tRNA, like the anticodon.
Yeah, exactly. So when this ribosome moves over one spot, it's going to be on the UUU, right? And because it's on the UUU, that means that this particular active amino acid can meet up with that UUU, which is AAA, right? And that one will join into the A site. So we'll say that the active amino acid is determined by the codon that matches specific anticodon. Okay, that makes sense. Then after that thing is in the A site, then the ribosome is going to move one location to the right. Whatever was in the A site will go to the P site. Whatever was in the P site will go to the E site. Okay, so the second thing that happens is we got to talk about the P site. So we'll say in the P site, the amino acid is detached from the tRNA and added to the growing polypeptide chain. Okay, so it's easy to remember what the A site is because the active amino acid enters in the A site, right? Active amino acid, all those things start with A, so they go into the A site. The P site is easy to remember because the P site is going to form the polypeptide chain. That's where the polypeptide is going to be held. P for polypeptide, right? And then the last thing is called the E site. So when the ribosome moves one more spot to the right, whatever was in the P site will now be in the E site. The thing that was in the P site was a tRNA that's now had its amino acid removed from it, right? And so we can say the tRNA that has had its amino acid removed will exit through the E site. E for exit. Okay? So, this process happens over and over and over and over again, and as it keeps happening, this polypeptide chain that's in the P site is going to keep getting longer and longer and longer and longer until it's the size that it's supposed to be. So if you look here, in this particular sequence that we've done, we've got AUG, whoopsies, that's not what I meant to do at all. We've got AUG, right? And that's the start codon. Then we've got UUU, right? That corresponds to the phenylalanine. We've got UAU. That corresponds to tyrosine. And then we've got UGA, that's a stop codon, right? So this is going to create a polypeptide then that's got methionine bonded to phenylalanine, bonded to tyrosine, and then that's the end of it. Now, that's not really a good example of a protein because it's only 3 polypeptides or 3 amino acids long, and that's super short. This is not a valid protein here, but if we had drawn more things in the center, then it would be. Okay, so what I wanted to get to here, what I wanted to point out is there are 4 codons, right? AUG, UUU, UAU, and UGA, but only 3 amino acids, right? That's because this last one, UGA, if we look on here, UGA is here, and it codes for this thing called stop, right? And stop is not a fancy word for some long named amino acid, right? It's actually stopping. So this is going to code not for a tRNA, but it's going to code for something called a release factor. Okay, so this whole process is called termination, and it's the 3rd step. So what happens in termination is eventually the ribosome is going to get to a stop codon. So eventually the ribosome will reach a stop codon. And we can say that stop codons code for release factors instead of tRNA. So these things called release factors, they're protein complexes, and what they do is they remove the ribosome from the RNA and they release the polypeptide from the ribosome. So release factors remove the ribosome from the mRNA and release the polypeptide from the ribosome. So those are your 3 steps. Step 1, small subunit attaches to the G-cap. It moves down the mRNA until it reaches AUG, which is the start codon. This methionine tRNA will attach to the start codon, and when it does, a large ribosomal subunit will attach to it. Once it's got the large ribosomal subunit, it's going to move down one codon at a time, so 3 nucleotides at a time. Whatever was in the A site will move to the P site. Whatever was in the P site will move to the E site. And the A site, new amino acids will enter. In the P site, the polypeptide chain will be held, and in the E site, the tRNA exits until it reaches this thing, which is UGA. And instead of having a tRNA go into the UGA spot, it's this protein called a release factor that breaks the small and large ribosomal subunit away, and it releases the polypeptide from the P site. Yeah?
2 questions. 1, these 3 steps are all part of what exactly? Just DNA?
No. These are all part of translation. So translation is the final step in protein synthesis. The first step in protein synthesis is transcription, and the second step is translation.
Okay. Do you think there's like, I don't know, some kind of like animated GIF or something online that would like do this animated?
I gotcha. We're going to it next.
Oh, sweet.
Wait, doesn't like that happen.
I know.
Did you get what you call a haircut, Mr. Teddy?
What I call a haircut, yes.
Hair trim.
All right.